whether they elicit feelings of fear or fascination, loving or loathing. It's hard to deny that spiders are incredibly successful animals. To date, around 50,000 species of spider have been catalogued, and there are doubtless many more that have yet to be named. And between them, they have colonized almost every habitat on land imaginable, right down to the forsaken corners, nooks and crannies of our own sterile abodes.
Ubiquitous in presence and beyond fathom in variety as the spiders may be, they, like all of evolution's manifold concoctions, from the mundane to the spectacular, must have started somewhere. So let us now delve into the fascinating facts and alluring mysteries of spider evolution, both their early origin and the diversification that followed.
The oldest fossil taxon that can be unequivocally identified as a spider is Paleothele montcoensis, which dates back to the late Carboniferous, around 300 million years ago. But if we're to examine the actual origin of spiders, then we're going to have to dive deeper into the past and broaden our scope beyond spiders themselves. Spiders are arthropods, an enormously diverse clade that single-handedly comprises over 80% of animal species known. Modern arthropods in their turn can be divided into two smaller groupings. On one side of the arthropod family tree, we have the mandibulates, which include crustaceans, insects, centipedes, and millipedes. On the other side of the family tree are the chelicerets, the clade that includes arachnids like spiders, scorpions, and mites, along with a couple other groups like horseshoe crabs, which despite their name are not closely related to crabs at all.
Chelicerets are highly diverse in appearance and lifestyle, but when one gets to the bottom of things, it soon becomes apparent that they are all variations of the same basic plan. Their body is divided into two main sections. The foremost of these is called the prosoma, which translates to mean front body. The rearmost portion is called the opistosoma, with an equally straightforward translation of rear body. In all chelicerets, the prosoma bears 6 pairs of appendages. The foremost pair are the cheliceret's namesake feature, the chelicerae, which are typically used for feeding. Next in line are the pedipalps, and the remaining 4 appendage pairs are usually referred to simply as legs, though in certain groups 1 or more leg pairs have been modified to serve alternative functions. When it comes to breathing, land-living chelicerets, which the majority of species are, typically employ one or both of 2 main features. One is a tracheal system, comprising a number of tubes that open to the air via tiny pores on the animal's exterior. The other is the book lungs, which consist of several layers of thin tissue stacked atop one another like the pages of a book, hence the name. The chelicerets have a deep fossil record, far more ancient than the oldest confirmed spiders, and their earliest known members can be traced back to a period that saw the opening chapters of many of evolution's greatest stories, the Cambrian, which began around 542 million years ago.
Our planet in the Cambrian was a very different place. The land was barren, inhospitable, and devoid of life. But in the oceans, conditions were much more favourable, and beneath the waves, life was flourishing. Many of the animals at this time were mind-bogglingly bizarre, but under the chaotic visage of this otherworldly ensemble were echoes of familiarity, early blueprints of features and body plans now widespread among animals living today. And it was in the Cambrian that the forerunners of the chelicerets, and by eventual extension the spiders, set down their roots.
One of these was a group called the Hybeliids. These were not true chelicerets per se. They lacked chelicerae for one, but still possessed features indicating that they were, at the very least, of close affinity to them. The configuration of the limbs on the front of the body matched the pedipalps and walking legs of the chelicerets proper. And even if the presence of chelicerae themselves was not yet apparent, the exceptionally well-preserved fossils of the genus Hybelia show a pair of small frontal appendages that, due to their position on the head, are regarded as likely precursors of true chelicerae. Moving on from the Habilids, we have another Cambrian oddity called Mollisonia. Strange as it may appear compared to its modern-day brethren, Mollisonia is the earliest known animal that exhibits all of the chelicerate defining features, and is thus the oldest true chelicerate known. Just like the Habilids, the limbs at the front of the body aligned with the typical chelicerate limb configuration, but unlike the Habilids, Mollisonia possessed true chelicerae, of appearance similar to those seen in many chelicerates alive today. The breathing apparatus of Mollisonia was also significant. Lining its opisthosoma was a series of book gills, alike to those that modern horseshoe crabs, some of the most primitive of all living chelicerates, still possess. And the book gills of these ancient aquatic chelicerates are considered to be the evolutionary precursors of the book lungs seen in land-living chelicerates like spiders. So, in the form of odd little critters like Mollisonia, the template for the body plan of spiders and all their relatives had been established way back in the Cambrian. But it wasn't until considerably later that chelicerates of a more familiar, spider-like appearance first arose, and to look at them, we must leap ahead from the Cambrian to the Silurian.
The Silurian period was a pivotal time in Earth's history, for it was during the Silurian that life, which for so long had been closely tied to the waters, began to expand its conquest over dry land. Plants and fungi spread across the Earth, creating in their turn a novel habitat for animals as well. By this time, some chelicerates had already become established as land-living, air-breathing animals, and though spiders themselves had yet to evolve, some of these Silurian chelicerates were already showing distinctively spider-like anatomy. Among these were a group of arachnids called the Trigonotarbids, which first show up in the fossil record around 420 million years ago. Trigonotarbids are not considered to have been direct spider ancestors, and were instead early relatives, but their appearance nevertheless provides clues about the origins of certain traits seen in actual spiders, with a major example being the modification of the chelicerae.
Chelicerae, in their original, unmodified state, were pincer-like as exhibited by the Cambrian Mollisonia, and indeed certain living chelicerates, like scorpions and harvestmen, still retain this old configuration. In this pincer form, the chelicera has two fingers. One is fixed in position, but the other has a hinge and is thus movable. The chelicerae of spiders are rather heavily modified from this base plan, forming not a set of pincers, but venomous fangs. Trigonotarbids, meanwhile, possessed chelicerae that, relative to the ancestral state, had an enlarged mobile finger and a reduced fixed finger, creating a jackknife-type structure, an anatomical intermediate between ancestral pincer-like chelicerae and the highly modified fangs of spiders. Furthermore, the spiders' closest living relatives, including amblypygids and vinegaroons, possess these same jackknife chelicerae. So even though Trigonotarbids were not always look on the bright side of life. So even though Trigonotarbids were not direct spider ancestors, they are still a useful piece in the puzzle of elucidating the spider's early origins, for they show that both a spider-like body plan and key features shared between the spiders and their closest relatives had already become well established at a time when life on land was still something of a novelty. And as we leave the Silurian behind and continue on our pilgrimage through prehistory, it's not too long before creatures of even closer affinity to true spiders appear.
The next stop in our journey is the period immediately following the Silurian, the Devonian, which saw the consolidation of life's earlier conquest of the land, and the continents, for the first time in the planet's history, were blanketed by vast forests. And it was in the Devonian that we see the first evidence of another major development in the origin of spiders. This is Attercopus, a 390 million year old arachnid, and one of the most vital discoveries yet in our quest to unveil the buried secrets of spider evolution. Anatomically, Attercopus was considerably closer to a proper spider than anything else we've covered so far. The two sections of the body are more clearly divided, and the chelicerae look largely similar to the fangs of true spiders, though still without evidence of venom glands. But most notably, Attercopus shows the earliest evidence of what would eventually become one of the spider's greatest innovations. The fossil remains of Attercopus preserve in remarkable detail a set of specially modified hairs called spigots, which are used to produce silk. This intriguing material, light and elastic yet shockingly strong, is perhaps more than anything else the key to the spider's success. Attercopus, however, did not yet have the means to utilize its silk to anywhere near the level of delicate finesse and mind-boggling sophistication that true spiders can. In spiders, the spigots are situated on a series of flexible appendages called spinnerets, which give them a great degree of control over their craft and are instrumental in the spider's ability to spin complex webs. Attercopus, unlike actual spiders, lacked spinnerets, with its spigots instead being distributed upon a series of flat plates on its underside. This would have restricted the arachnid's usage of silk to fairly basic, rudimentary functions, perhaps to protect its eggs, or as a simple adhesive to help hold the structure of a burrow. And that in turn raises one of the most fascinating questions in spider biology. How did spinnerets appear?
To answer this, it's probably best we take a few steps back. As aforementioned, all chelicerates have 6 pairs of appendages attached to the prosoma, the front of the body. That's simple enough, but to discuss the potential origin of spinnerets, we have to shift our focus to the rear portion of the body, the opistosoma. The opistosoma does not, in many chelicerates, including ancient spider relatives like Attercopus, appear to possess any obvious appendages at all. But that wasn't always the case. If we revisit those Cambrian proto-chelicerates, we can see that they had an abundance of appendages attached to the rear of their bodies. Furthermore, the horseshoe crabs, which, due to how ancient they are and how little they've changed over time, can be excellent exemplars of primitive chelicerate anatomy, still have a similar set of opistosomal appendages. Based off this, it's reasonable to conclude that the ancestral chelicerate body plan did have appendages on the opistosoma, and that these were subsequently reduced, lost, or modified beyond recognition in later, more derived chelicerates, including the ancestors of spiders. However, when a feature is lost over the course of evolution, the genes that coded for that feature often remain in the organism's DNA, but are no longer expressed. And a genetic mutation is all it can take to cause a re-expression of dormant genes, and consequently a reappearance of lost features. In Attercopus, the flat plates over which the arachnid's silk-producing spigots were spread are thought to have been the remnants of lost appendages, as appears to be the case for the similar plates we see in scorpions. A re-expression of the relevant genes in an ancestral spider would have caused these lost appendages to reappear, now bearing the spigots that once sat upon the plates, thus giving rise to the first spinnerets. More recent research analyzing the genomes of spiders and other arachnids has since added further depth and credibility to this proposal. Evidence has been found of large-scale mutations known as gene duplication events, a major factor in evolutionary change, occurring early on in the spider's evolution, and such events could have facilitated the reactivation and re-expression process that gave rise to spinnerets.
The appearance of spinnerets, though undeniably of great significance, wasn't the only change that needed to happen to bridge the gap between an Attercopus-type body plan and that of a true spider. Primitive spider cousins like Attercopus also had a long, whip-like tail, alike to that possessed by modern vinegaroos, which gives reason to suspect the spider's direct ancestors were tailed too and lost this feature later down the line. But what change occurred first, the appearance of the spinnerets or the loss of the tail? Well, one small but significant creature might provide a near-perfect answer. Meet Chimerarachne, a 3mm long arachnid whose remains were found encased and immaculately preserved in amber, which is fossilized tree resin. Chimerarachne at a glance resembles Attercopus, but if you decide to examine its rear, which in the field of paleontology is a lot more socially acceptable, then it soon becomes apparent that Chimerarachne had something very notable: well-developed spinnerets. And since this bizarre arachnid still possessed the long tail seen in Attercopus and its ilk, it's thus reasonable to conclude that the evolution of spinnerets preceded the loss of the tail. Chimerarachne had something else of significance too. A major defining feature of spiders is the modification of the pedipalps, the second pair of appendages behind the chelicerae, into specialized reproductive structures, at least in the adult males. And this same trait is clearly visible in the fossils of Chimerarachne. So between the pedipalps and the spinnerets, it's evident that Chimerarachne was a very close relative of true spiders. But we can also say for certain that it wasn't a direct ancestor to them, and the reason we know that is its age.
The remains of Chimerarachne date back to about 100 million years ago during the Cretaceous period, some 200 million years younger than the oldest fossil spider known. The age of Chimerarachne might therefore appear confusing, but there's a pretty simple explanation for its seemingly anomalous position amid our ancient arachnid chronology. And that is that Chimerarachne was a late surviving member of a primitive group that was closely related to the ancestral spiders. And that makes this tiny arachnid all the more notable a discovery. For Chimerarachne provides clear evidence that the spider's ancient cousins weren't mere prototypes that fell into extinction as soon as proper spiders appeared on the scene. They were successful animals in their own right, having continued to exist alongside their more modern and familiar relatives for at least a further 200 million years. Now, finally, we come to the first actual spiders. As mentioned earlier on, the oldest fossil spider yet discovered is Paleothele montcoensis from the late Carboniferous about 300 million years ago. Though given the incompleteness of the spider fossil record, it's likely that spiders go back a fair bit further than Paleothele itself, but have not left any known remains in the fossil record, with multiple phylogenetic analyses estimating the origin of spiders to be somewhere within the preceding Devonian period. Paleothele belonged to a group of spiders called the Mesothelae, and we don't have to imagine what the Mesothelae may have been like, because these, by far the most primitive of all spiders, still cling on to existence by a thread.
Today, the Mesothelae account for only a minuscule fraction of spider diversity, comprising less than 100 out of the tens of thousands of spider species alive, and are exclusively found in Asia. Since their distant origin, the appearance of the Mesothelae has changed little, and they still retain very primitive features not seen in any other spiders. Perhaps the most obvious is the well-defined segmentation and armor plating on their opisthosoма, a trait they share with their prehistoric cousins like Attercopus and Chimerarachne. Less immediately visible is the group's namesake characteristic, the position of their spinnerets. These are situated roughly in the middle of the opistosoma, hence the name Mesothelae, which means middle teat, with teat in this case referring to the spinnerets. The Mesothelae's usage of silk is relatively simple. They use it to line their burrows, and in some species, to create a series of radiating trip lines that alert the spider to the presence and whereabouts of prey wandering around outside.
The Mesothelae may have scarcely changed since first they appeared, but the same can't be said about the other major division of spiders, the Opisthothelae. Now, just from the name, you should be able to tell how the Opisthothelae may differ from their more primitive counterparts. As we've touched on, the prefix opistho means rear, and thelae refers to the spinnerets, so it should be clear from the name that the Opisthothelae, unlike the Mesothelae, possess spinnerets at the rear of their bodies. This is a somewhat more optimal position for web making, not least because the spinnerets are less obstructed by the spider's own body, and that allows the Opisthothelae to construct more expansive and complex dwellings than their ancient relatives. The Opisthothelae in their turn are divided into two groups. One of these is the Mygalomorphs, which include tarantulas, trapdoor spiders, mouse spiders, and funnel webs. The other is the Araneomorphs, which include, well, pretty much everything else.
The Mygalomorphs exhibit less overall change from the ancestral spider body plan. Though more derived than the Mesothelae, they still retain some of the features seen in their more primitive cousins. The fangs of Mygalomorphs are held roughly parallel to one another and strike downwards. Their respiratory system is also similar to that of the Mesothelae, consisting of two pairs of book lungs. This breathing apparatus is somewhat inefficient and susceptible to drying out if the spider stays too exposed, which places restrictions on the sorts of habitats and lifestyles available to Mygalomorphs. To stay safe and sheltered from desiccation, Mygalomorphs are almost exclusively burrowers, typically living on the ground, though some, like certain species of funnelweb, may inhabit trees as well. The imperfections of their breathing system also render them incapable of sustained, high-speed activity, so a life of actively foraging and chasing down prey is off-limits to them. Instead, Mygalomorphs are masters of ambush, operating from secluded hideouts and patiently waiting for victims to stray within reach, before attacking with a short but tremendous burst of speed and power.
Mygalomorphs hold a couple of the spider world's most notable records. For one, they include the biggest spider alive, and for that matter, the biggest spider known to have ever lived, Theraphosa, an enormous tarantula native to South America. Another Mygalomorph, Gaius from Western Australia, holds the equally memorable title of being the longest-lived spider on record, with one female having a documented lifespan of 43 years. And with over 3,000 living species across a broad geographic range, the diversity of Mygalomorphs is certainly respectable, but it scarcely holds a candle to the other subdivision of the Opisthothelae, the Araneomorphs.
The Araneomorphs comprise a whopping 93% of spider species alive, and include just about every commonplace and familiar spider you could possibly think of. Amid this diversity is a suite of evolutionary innovations not seen in any of the more primitive spiders we've covered thus far. One of the most prominent features of the Araneomorphs is the orientation of their fangs. Unlike the downward-striking fangs of the Mygalomorphs and Mesothelae, the fangs of Araneomorphs point inwards towards one another and bite in a pincer-like motion. The art of web making is also something the Araneomorphs have taken further than any other spiders, and central to this is the further development and specialization of their spinnerets.
In the Mygalomorphs and Mesothelae, the different pairs of spinnerets may vary in size but are otherwise largely the same. On the contrary, the spinnerets of Araneomorphs are more diverse, with an individual spider possessing the means to spin multiple, different types of silk. The breathing apparatus of many Araneomorphs is also modified to an extent far beyond that of their ancient relatives. With the exception of some of the most primitive species, which still retain the ancestral configuration of 4 book lungs, most Araneomorphs have only 2 book lungs, with the other 2 being replaced by a tracheal system, which is comparatively more efficient at supplying oxygen to the animal's body and less vulnerable to drying out, making it possible for these spiders to live out in the open, fully exposed to the elements. This tracheal system is thought to have initially evolved from the book lungs, and since then become progressively more complex and expansive. And nor was the development of the tracheal system a one-off innovation either. Genetic studies indicate that across the Araneomorphs, a tracheal system arose from ancestral book lungs 6 separate times, each in a different Araneomorph lineage, setting the stage for the great diversity of breathing systems we see among these spiders today. But what factors could have compelled the Araneomorphs to, in multiple instances, develop this novel, complex, and more efficient mode of breathing? Well, it's possible that it may be linked to the Araneomorphs' advanced web spinning. The close proximity of the tracheae to both the silk glands and the muscles operating the spinnerets indicate that they play an important role in supplying energy to these particular body parts, and it's been proposed that the evolution of the tracheae may be linked to the evolution of more complex webs, with the tracheal system developing to fulfill the elevated energy demands of an increasingly sophisticated silk-spinning apparatus. The earliest fossil evidence of both the Araneomorphs and the Mygalomorphs dates back to the Triassic period, but phylogenetic analyses indicate that these two groups both arose considerably earlier than what their rather limited fossil records would suggest, perhaps around the beginning of the Permian or even in the Carboniferous. These same analyses indicate the Araneomorphs underwent much of their diversification during the Mesozoic era, and by the Cretaceous period, many familiar modern-day Araneomorph families had become established.
But though the types of spiders present in the Cretaceous may have, to a large extent, paralleled what we see in the present day, their relative abundance and diversity may have been quite different. Today, the world's spider diversity is overwhelmingly dominated by two Araneomorph clades. One is the Araneoidea, which includes most of the conventional orb weavers and their close relatives. The other is the RTA clade. The RTA clade is a huge group of spiders, containing over 21,000 species across more than 30 different families, all united by the presence of a distinctive backward-facing projection on the male pedipalp. This is called the retrolateral tibial apophysis, abbreviated as RTA, and is, of course, the clade's namesake feature. With some exceptions, like this Dendrolycosa, members of the RTA clade tend to have little to no reliance on webs to ensnare their prey, instead opting to hunt it directly, either through ambush or active pursuit.
The origins of both the Araneoidea and the RTA clade are estimated to have occurred as early as the Jurassic period, but no fossils of RTA clade spiders are known from the entire Mesozoic era, and the Araneoidea only constitute a small portion of spider remains uncovered from this time. Instead, the dominant spider groups from this time appear to have been the Synspermiata and the Palpimanoidea, both of which are still present today, though exhibit comparatively limited diversity. However, once we move forward from the Mesozoic era to the Cenozoic era, the composition of spider species in the fossil record becomes much more reflective of what we see today, with the Synspermiata and the Palpimanoidea being greatly diminished, and the RTA clade and Araneoidea forming a clear majority of spider species. What this indicates is that, though they were both present, the RTA clade and Araneoidea constituted a relatively minor portion of spider diversity through the Jurassic and Cretaceous periods, and that between the Cretaceous and the Cenozoic, the dynamic between these two clades and the then much more dominant Synspermiata and Palpimanoidea was effectively turned on its head. But what could have brought about this dramatic shift? Well, we can't say for sure, but it is worth mentioning that between the Cretaceous and the Cenozoic, a somewhat notable event occurred. The Chicxulub asteroid impact, which brought an abrupt end to the Cretaceous period 66 million years ago, is best known for wiping out the non-bird dinosaurs. But this cataclysmic event also had devastating consequences for many other living things, great and small. Mass extinction events can often be the catalyst for major turnovers in biodiversity. When an established, dominant group gets diminished or completely wiped off the map, other groups, now largely free of competition, can evolve and diversify in their place. And this may have been the case for spiders, with the dominant spider clades in the Cretaceous potentially suffering heavy losses as a result of the Chicxulub impact, thus providing an opportunity for the RTA clade and the Araneoidea to take their place. But it's worth mentioning that, though we have fossils of spiders from both the Cretaceous and the periods that followed, there are none yet found that date back close enough to the event, either before or after, for us to be able to properly test the role it may have played in shaping spider evolution. But whatever the cause, it does at least seem clear that, not too long after the end of the Cretaceous, the world's spider diversity was more or less in line with where it is today. And with that, we finally come to the end of this deep dive into the ancient history of some of the planet's most fascinating predators, and the references are linked in the description for your perusal. If for some reason you aren't yet completely fatigued from hearing me ramble about spiders and want to listen to even more of it, then take a look at this video about the Jurassic Mygalomorphs. And if prehistoric insects are more your fancy, then head over to this video about the huge and fearsome Gigatitan. And don't forget to subscribe so you don't miss out on any future content. Thanks for watching, and I'll see you on my next adventure.